So welcome back to another exciting tour, no doubt, of some of those Bi-12 biochemistry objectives. This one asks you to demonstrate a knowledge of synthesis and hydrolysis as applied to organic polymers. Right off the bat, notice you're talking about two objectives. Whatever this synthesis thing is, whatever this hydrolysis thing is, as applied to organic, and somewhere I am sure you have come across the term organic. In this context, we're talking about hydrocarbons, carbon, hydrogen, and oxygen. So these are the building blocks of the polymers, those wonderful macromolecules that you already are very familiar with. Right. Do you know them all? Again, the four polymers that you are responsible for knowing are the nucleic acids, the lipids, the proteins, and the carbohydrates. A diagram you're going to see often is something like the one shown below here. The reason it's shown and used so much in this unit is it really is a good way of determining your understanding of the objective. Even without going into detail here, you can already tell just by looking at the diagram that if you have a bunch of small molecules, these monomers, if you like, so this could be glucose, 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 the monomers of the carbohydrates. If you remove something from those, and we won't tell you what that is yet, although I think you have a pretty good idea, you're going to end up forming not the monomers, but the polymer, the actual macromolecule itself. So in the case of if this was glucose, this has got to be a carbohydrate like a disaccharide or a polysaccharide. At the same time, if you start with the macromolecule and you add whatever this substance is, you're going to end up going full circle and returning to the starting point. So, synthesis. Word itself in this context is to build up. It means to take the small building blocks, the monomers, and from that produce something much larger. So again, the four macromolecules, you need to be able to demonstrate this for each of them. In the case of carbohydrates, you know what the monomer is. It's going to be glucose. So if you take two glucose monomers, combine them chemically, you're going to get a larger molecule, in this case a disaccharide, maltose. And always, always, always in a dehydration reaction, you're going to end up with water. So just to clarify one thing, you're going to sometimes hear synthesis called dehydration synthesis. It's probably a better term because it describes what is actually happening. You're removing water from the reactants and it shows up as a product. So always in dehydration synthesis or condensation synthesis, water is a product as well as a larger molecule than you started with. So that would be a word form of synthesis. You also, of course, will be shown chemical symbols for those reactions, and by now, again, hopefully you don't feel too uncomfortable with seeing glucose, C6H12O6. Two glucose units combine chemically, they bond to form a disaccharide called maltose. Don't worry about the 1,4 glycosidic linkage, that's simply what that bond is called. But it's a good idea to notice where the water is actually coming from here. You had a hydroxide group here, the OH. You had an H on this hydroxide group, HHO. Two H's, one O. That is, of course, water, so that's why it's a product. So that would be synthesis as applied to carbohydrates. What about proteins? Proteins, as you know, are basically combinations of amino acids. And amino acids plus amino acid is going to get you a small protein called a peptide, and again you're going to get water. So word form and then the actual chemical diagram form. One amino acid showing the OH, another amino acid showing the H. This is where the water is going to show up as a product on this side of the reaction. And again, two amino acids just are dipeptides. What about lipids? Lipids, of course, are these three long fatty acid chains joined to a 3-carbon molecule called glycerol to form a fat and again water. This is just a symbolic representation because the fatty acid chains can actually be very long, but always, always, always when you form a fat, you are going to have to combine 3 fatty acids with a 3-carbon molecule called glycerol. And because each of those glycerol molecules has 3 OHs, and they're taking 3 H's from the fatty acids, guess what you get? You get 3 water molecules as a product as well. Then the last one would be the nucleic acids. Nucleic acids are formed by dehydration synthesis of nucleotides. A nucleotide plus a nucleotide gives a nucleic acid like DNA or RNA, and again water. The diagrammatic representation really shouldn't freak you out. This is simply a nucleotide, phosphate. This would be the PO4 group, the phosphate. This would be the sugar. And there's only 2 possibilities here. If this is DNA, this is deoxyribose, and then this could be a base like adenine or guanine or cytosine or thymine. But nucleotide 1 plus nucleotide 2 combine chemically, and again there's a fancy bond that joins them called a phosphodiester linkage, which you can promptly forget, and form water. So back to the original diagram we were shown, which you will see in various forms. You should be able to now look at a diagram like that and say, okay, what happens if I take the monomers for, say, lipids? They would be fatty acids and glycerol. I remove water, I'm going to form a fat. If I add the water back, I'm going to break down the fat back into the starting monomers, the fatty acids and glycerol. So in your textbook, you'll see in general a diagram like so that shows that for any monomers, if they undergo dehydration synthesis, they're giving off water as a product and combining those monomers into a large molecule. At the same time, if you take the large molecule and you add water, we call that hydrolysis. Speaking of hydrolysis, you'll recall that the objective isn't only to demonstrate a knowledge of synthesis, but it is also to demonstrate a knowledge of hydrolysis. Hydrolysis is kind of a nice word because hydro immediately tells you you're dealing with water. Hydroelectric. Lysis, on the other hand, always means split or to break apart. So this is telling you already that something is being broken apart upon the addition of water. And that is, of course, called hydrolysis, and it simply means breaking apart. Breaking up. Not any sort of romantic connotation here, but breaking up, and I could write more, but the idea is breaking up in the presence of water. So again, the 4 macromolecules, you simply reverse the reactions we've already looked at. You now take the product plus the water to get the original reactants. So we take a disaccharide like maltose, add water, you're going to get the 2 monomers, glucose plus glucose. And again, don't be afraid of the diagrammatic representation of that. That is 1 molecule, so that is a macromolecule. If you see water on the reactant side, this is hydrolysis because now you end up with the 2 monomers. And again, without showing you the actual molecules, I'll just quickly run through the other 3 macromolecules. For proteins, peptides, if you add water to them, you're going to get the monomers, the amino acids. If you take fats, of course lipids, add water to them, you're going to get the 3 fatty acids and glycerol, the monomers that make them up. And of course, nucleic acids like DNA or RNA undergo hydrolysis to form the nucleotides that make it up. So I'm going to leave you with the diagram that we simply started with. You could do yourself a favor and just pause the video and go through the 4 macromolecules and just make sure that regardless of which one, you can tell me what substance A is, substance D, substance C, and again, I think by now you realize that substance D is always going to be water. Substance A is going to be one of the monomers for any one of those 4 macromolecules, whether it's glucose in the case of carbohydrates, could be amino acids in the case of proteins. What would you get as a macromolecule?